Yellow fever mosquito, she can lay a few hundred eggs over her lifespan. Malaria mosquito is probably about the same, a few hundred eggs. So this means that, you can have a bad problem become much worse if there's a lot of places for mosquitoes to breed.
This is Fissionary, a show exploring how nuclear powers your world. I'm Mary Carpenter.
And I'm Jordan Houghton. Let's jump in.
Welcome back to Fissionary. This season, we're asking one big question. What powers wonder?
And today's answer, innovative genetic tools, or more specifically, how pairing nuclear science and insects have the ability to improve global public health. Mary? I don't even know where to start. Our guest today, Dr. Zach Adelman, professor at Texas A&M, one of the most fascinating guests I think we've had. I learned so much. I learned so much that I don't even know if I want to know what I know now after having this conversation, because I am infinitely more aware of all of the ways insects are like seeking to destroy the life we know and love behind the scenes. And the scientists are there using nuclear science to thwart them, which is great. But holy moly, this was an eye-opener.
Yeah, I have obviously never been a big fan of bugs. I've always been very interested in bugs. I took an entomology class in college, which was one of the classes I really remember because our teacher was so interesting. Shout out to Dr. Espley. But I've always been interested in bugs, but I never knew how cunning and smart they were until today.
Well, and for our listeners, we really came into this thinking we were going to talk about mosquitoes because, we were planning this. We're like, wait, nuclear science is helping get rid of mosquitoes, which are the deadliest animal in the world, causing deaths from so many different diseases, but also just a horrible nuisance. Nobody likes to be outside in the summer getting bit by mosquitoes. And it's just, so we're like, we're going to talk about this, and learned so much about other insects that nuclear science is helping control, from the screwworm fly to fruit flies, and their grave impact on our food ecosystem.
Yeah, it's terrifying to think that if they weren't using these techniques and technologies, that our food supply would be drastically impacted. You guys will hear from Dr. Adelman in the episode, but I mean, we wouldn't have the fruit supply we know. It just wouldn't exist. And meat would be heavily impacted. We wouldn't be able to just go to the store and buy a steak. It would look a lot different.
And he makes a great point about the cost of the meat and the fruit, if we were able to even get any, how much it would cost, because the supply would be so short because of the havoc that these insects wreak on populations of cattle, fruit, trees, etc. I think we should just take our listeners on this journey with us. Come on in.
Yeah, let's dive in. Dr. Zach Adelman is a geneticist and professor of entomology at Texas A&M University. His work focuses on developing new tools to control mosquito populations and reduce the spread of diseases like dengue, malaria, and Zika. Thanks so much for joining us today, Zach.
Thanks for having me.
Zach, we're really excited to have you. We're talking about this season, What Powers Wonder. A bug-free world is something that is wondrous to me. But I want to ask you, in your work, what sparks the sense of awe, curiosity?
I'm really interested in kind of the interplay between our genome and the world that we live in, right? So how does the genetic material that's wrapped in our DNA, how is that read, encoded? How does it change over time? And how does that yield all the different forms of life on Earth? To me, that's endlessly fascinating.
That's cool. Okay, so let's get into what we're here to talk about, mosquitoes. One of my least favorite topics, but I'm very excited to talk about what we're going to talk about today. So mosquitoes are often seen as simple pests, but your work really reveals how sophisticated they are as a species. What makes them so good at spreading disease and so hard to manage?
All right, so first things first, right? What makes it so hard? What makes it so hard to control it? And kind of the biggest misconception I think anybody has about mosquitoes. If I asked you what a mosquito was, you might describe something that flies around and makes a whining sound as it goes past your ear and then bites you and then get big red welt afterwards, or maybe you'd get sick if you got something. But in reality, there's more than 3,000 known species of mosquitoes. Probably there's even more than that we haven't even discovered yet, right? And that number is not just, oh, wow, there's a lot of different species. It's that they have been around for a really, really long time, and they're really, really genetically diverse. If I put a malaria mosquito next to a dengue mosquito, and I showed them to you under a microscope, because they would both look about the same size and they're about the same color, they look very different to me. But I'd say, hey, how different are these mosquitoes? And you're like, yeah, it's another mosquito, but they're more than 200 million years separated evolutionarily, right? And so that would be the equivalent of me looking at a person, like I'm looking at you guys right now, or myself, and a duck-billed platypus and saying, they're both about the same.
Interesting.
So once you understand how genetically diverse mosquitoes are and how ancient, then you can understand how difficult it is to try to have any means of controlling them, because what works against this species will not work against another one or another one by one. They all have their own habits, ecologies, preferences, susceptibilities, lifespans, any behaviors. Everything is going to be pretty unique, all these different groups. So you're not trying to control one thing, and the strategies have to be different too. The ones that cause the most trouble for us, right, out of those 3,000 something species, only a handful, a couple dozen, probably cause almost all of the human disease transmission. These are the ones that are really well adapted to people. They'd much rather feed on people than anything else. And they've gotten really, really good at it. Again, when they first started blood feeding, where were we? We were the progenitor of whatever the original mammals were going to be. And they've been following us throughout our evolution, figuring out ways to feed on us without us detecting them, right? So they secrete all kinds of stuff into your skin, react to, but it also makes it so you don't feel the bite, so the blood flows easily, so it doesn't clot. All these different strategies, they can get in and get out without being spotted. And so they can detect you from distance. They can hone in on all kinds of signals that you're giving off, chemicals that are coming off of your skin, your body temperature, the CO2 you breathe. They can see the heat that you're generated through infrared. They can just hone in on these cues from a long distance, and they know how to get in, they know how to get out. And we're just still trying to catch up with all of their tricks.
Wait, like the Predator?
Yes, like the
movie
Predator.
They see it.
Yes, exactly like that. There was a really great paper from a group out of UC Santa Barbara, Craig Montella's group, who showed they actually do see infrared.
This is so much worse than I thought it was.
I know, it really is. I came into this and I'm like, hate mosquitoes, they create a nuisance, they create disease, and now I'm like, this is grave. This is really dire.
Yeah.
Is it true mosquitoes are the deadliest animal on Earth?
Through the transmission of malaria, then yes, because malaria causes so much mortality, going on 500,000 deaths.
Wow.
They're all similar to each other, right?
Okay.
The one mosquito and another is the same, and they're not, right? So you have the Culex mosquito that you might find in your house. It's small and brown. You have a tiger mosquito that you might find in your backyard, and you're like, oh, it's one mosquito, it's another. I react to mosquitoes, or mosquitoes love me. But different species will make you react very differently. You might react to some species, but not to others. And you might be really attractive to some species, but not to others. And so this overgeneralization that there's only one thing out there is probably the biggest misconception.
So you've shared that male mosquitoes are expert trackers of female mosquitoes, and that evolution actually does the work. Can you walk us through how that works in the wild or in controlled release programs?
Sure. So males, really, they don't drink blood, right? So that's another maybe misconception that people have. Right, so male mosquitoes are not interested in feeding on you. They don't need your blood. They can't even do it. Their mouth parts are physically not able to penetrate your skin. So they just need to find females. That's their one job in life. And so there's really two big strategies that different mosquito species use. And again, knowing that there's actually a lot more nuance here, but males that are hunting females based on their sound, right? So have you ever had a female mosquito when she's trying to feed on you and she'll just go past your ear, right? And you hear this really high pitched kind of scream or squeak. Have you guys heard that before?
Yes.
Oh, yeah.
Yeah, exactly. So the male can hear that sound from really far away. right? And he is attuned to try to find that exact pitch, because based on the species, they'll always use the exact same pitch because their wings are the same size, they beat at the same frequency. And so he can tone in on that frequency and try to find her to try to mate. And so that's really great when you're talking about a sterile insect technique program where you're releasing males to then go and find those females, because they can hear that from much, much further away. For us, they have to be pretty close to our ear to be able to hear it, but they can hear it from a much long distances. For other mosquitoes, like malaria mosquitoes, the males don't seek females at all. They actually form what's kind of like we call a mating swarm. So he'll get dozens or hundreds of males that will all kind of find each other. And they form this big kind of cloud of male mosquitoes. And presumably you guys have walked through somewhere where there's a cloud of gnats, and you walk through it, you're like, oh man, a bunch of those gnats just went in my. That's the same kind of mating swarm that they use, right? So the females, because you have this swarm of all these males and all their wings are beating at the same frequency, it amplifies the sound. So the females can find a swarm and they fly into it. And then there's a big competition from all the males in the swarm to see who can grab her first.
Sounds like my worst nightmare. A giant swarm of hundreds of mosquitoes.
Yeah, but male mosquitoes, so they don't bite. They don't feed on you.
This is wild. Okay, so you mentioned sterile insect techniques. Can you give us a quick explanation of what that is and how it's used?
Sure. So insects, right, they're a problem because they reproduce fast. They reproduce a lot, right? So a single yellow fever mosquito, she can lay a few hundred eggs over her lifespan. Malaria mosquito is probably about the same, a few hundred eggs. So this means that, you can have a bad problem become much worse if there's a lot of places for mosquitoes to breed. And if you ever live in a place that has a lot of rain and then you get a lot of mosquitoes coming out, you know how quickly they can amplify. Their lifespan is pretty short. And so, they only live maybe a couple weeks in nature. And so over time, if you just rely on them, just their lifespan, they're just going to be gone in a couple of weeks. So they have to continually reproduce, and you're always getting these new generations. And so this makes them susceptible to a strategy where if you interrupt their ability to reproduce, you can massively affect the population, right? So if male mosquitoes, when they mate with a female, the female will then store that sperm that she collects from the male, and she's done. She has a special organ in her body where that sperm is kept alive. And then whenever she wants to fertilize her eggs, when she's laying them, she will just draw upon the stash that she keeps of these sperm over the course of the many weeks that whatever week she's alive. And so if she mates with a male that had been irradiated and is producing all inviable sperm or dead sperm, she doesn't know that. And so she won't mate again. And she just has an organ full of dead sperm. And when she tries to fertilize her eggs, it doesn't work. They don't form embryos. They don't develop into new mosquitoes. And so she'll live out her life, which is only going to be another week or week and a half or something like that. And then she can't produce any progeny. And if you do that over enough time, then you can massively decrease the population.
So tell us how nuclear science and radiation plays a part in the sterile insect techniques.
Well, we really couldn't do it without it, right? So this is the linchpin of how you get this kind of sterilization. How do you get these males that are going to be infertile? And it's been the use of, isotopes. Primarily right now, I think what is used is cobalt-60, which produces nice gamma radiation that can sterilize the mosquito. They're treated when they're basically pupae, which is the metamorphic stage between the swimming larvae and the flying adult, right? And so just like you have a butterfly, it has a little cocoon. Mosquitoes form the same type of structure, but they can still swim around while they're doing it. But if you irradiate them at that time, they basically can still complete development. They can still become adults. They still look morphologically normal, but the radiation has damaged their developing germline to the point that they're sterile.
So where did this idea come from? Who first thought, what if we use radiation to control insects?
It wasn't until, the 1920s, we started out with Hermann Mueller, who was a Drosophila biologist, who realized that, or first established basically that principle that if the more irradiation you treat these flies with, and he was using X-rays, but it ended up being similar for gamma rays or other types of radiation based on the decay, the more chromosomal damage you had, the more lethality and sterility you had in the chromosomes of those flies. It was Edward Knipling, who was an entomologist for the USDA. He was also a former Aggie, so he graduated from Texas A&M, from the department that I worked for now, who was studying screwworm and who knew a lot about screwworm biology. And he thought that, just treating cows and treating cattle, because the adult screwworm flies, they lay their eggs in wounds or orifices around ruminants, and then the larvae burrow into the skin. They make these giant wounds and eventually result in the death of the animal. But he spent all his time treating these cows, and it's like, man, if we could just get rid of these female flies before they get to the cattle, it would be way more effective. So he knew he wanted to stop them from reproducing, but he didn't know how to do that. And so he came across Muller's work, and then he put the two and two together and really thought, oh yeah, here we have something with radiation that can cause sterility if you have a high enough dose, and I want sterile flies. And so he got started with a few other folks there at USDA and got that going.
So, how successful has sterile insect techniques been on mosquitoes in addressing diseases like malaria, Zika, dengue?
There's been a lot of successes, and sterile insect technique has done really, really wondrous things for a lot of agricultural pests. It has been used for mosquitoes, but some of the trials have been successful. A lot of them have shown reductions, but this problem is scale and immigration. So if you're doing a trial in one small area and you basically reduce the population in that area, but mosquitoes can fly in from neighboring areas, they come back and they reproduce so fast that the population comes right back again. And so we really haven't seen a program at the scale that was achieved with the screwworm in order to get rid of it here. And so far, sterile insect technique has not been kind of a major force for controlling different mosquito species. There's been some successful trials. There's been a lot of what I would call smaller scale work, which is still really big, like citywide or kind of, maybe a county or things like that, but not across huge geographical spaces yet.
Talk about how they handled sterilization techniques in the screwworms geographically.
Yeah, so that started with some really good proof of concept trials on a few islands in the early 1950s. And it was the trial in Curacao that really convinced the producers that this was going to be something successful, right? So they were able to eradicate the screwworm off an island in the Caribbean through inundative releases of these sterilized screwworm flies. And the folks in Florida saw that because the Caribbean is right there next to their doorstep. And so the screwworm, where it was in the U.S. was in Florida, a little bit on the eastern side of the southeastern U.S., and then mostly in Texas and whatnot. And so the Florida folks saw those trial in the Caribbean and said, we need this now, make this happen. And so then they had the will to kind of scale up, and they started eradication to Florida, and they were able to get it out of Florida and the southeastern U.S. And that really caught the attention of the folks in Texas. And then by the mid-1960s, they had pushed it all the way out of the U.S., out of down the southern border, where they held it for a few years, and then realized that, they still had to release so many flies, 100 million flies per week along the Texas-Mexico border every year to stop them from coming north again. And that's when they really started to think, okay, we've done this at such a large scale already. Let's think bigger. Right. And so this required a huge international collaboration. So we worked very closely with the governments of Mexico and then subsequent all the governments of the Central American countries, producing these flies first in Texas, then in Mexico. And then as we chased them down year by year by year, starting in the 1970s, all the way through 2001, when it was declared eradicated from Panama. And then for the past 20, 25 years or so, we've held that line with a factory in Panama producing 20 to 30 million flies per week, releasing them on the border of the Isthmus of Panama to prevent them being reintroduced from Colombia.
So they release that many weekly?
Weekly, yes.
Oh my gosh.
Okay, so Mary and I obviously very focused on the mosquitoes, but we're talking about the screwworm fly. Are there other agricultural pests that this program has helped?
We basically would not have any fruit in this country if it weren't for the factories that are churning out millions and tens of millions of Mediterranean fruit fly and Mexican fruit fly along the California and Mexico borders right now. And those are released all the time to keep these things from reinvading. And sometimes they show up and they release a ton, make sure that they don't spread, and they eradicate it back again. But basically, our food supply is kept safe from these releases. And these programs started in the 1960s or 70s, and they have been going ever since. And these factories every week are churning out. There's a facility in Guatemala that can produce 1 billion flies per week for some of these Mediterranean fruit fly programs to protect Guatemala, Mexico, and the U.S. from these. And the U.S. has its own plants that produce Mediterranean fruit fly, Mexican fruit fly, and these are released all the time. And we have fruit because, and these flies can destroy 90% of what you've got, right? And so they don't eat all of the fruit, but they might feed on it or they might oviposit near it. They just damage the fruit enough that fungi will grow or other things will grow and destroy the crop, right? So if they feed a nibble here, and they nibble on every little piece, then all of a sudden it's all gone, right? So yeah, so we have fruit in this country because these programs have been running nonstop for 50 years or more.
That's unbelievable. So clearly we're seeing success stories. Do you ever see pushback to these tactics?
I don't think I've seen any pushback to sterile insect technique. It's nice because in terms of like compared to a pesticide where you can spray a pesticide over an area and basically just kill every possible insect, including all the ones you don't want to kill, right? All the beneficial insects, all the friendly ones, all the pollinators. This is really, really, really specific. These males are only going to go after females of their own species and no one else. So yeah, I haven't seen any pushback against sterile insect technique. It's listed as there's really nothing about it.
You just mentioned pesticides, which was going to be my next question about how this compares. And I think the thing that you just mentioned is probably the biggest difference between the two, and that you're not just killing everything, which includes bugs that are beneficial. Are there any other comparisons between the two? Like, how do the economics shake out? Is it more cost effective to do this ultimately?
So it depends on what your starting population is. And looking back at the original, description of the sterile insect technique and how it works best, it tends to work best. Again, you need these really high ratios of factory reared males compared to wild males, right? So if there's a certain number of wild males out there, you need to be rearing 10 times more than that or 100 times more than that, right? So if the population of the insect you're trying to control is really, really, really high, it makes it so much harder to produce enough males to get those ratios that you want. And so it tends to work best when you are able to reduce that population or if it's naturally low. So sometimes you might have to come in and use pesticides first to get the population kind of reduced to the point where sterile insect technique can really, you can get the ratios that you want. So they're not always exclusive, but for definitely when you have an introduction, right? So a lot of the sterile insect technique programs we still use in this country are for when we have an introduction or we spot the Mex fly, the Mexican fruit fly, or the med fly in Texas or in California. The populations can be very, very small because it just showed up, right? It just got imported in. So that's a great time to hit with sterile insect technique because it works in your favor. Whereas pesticides are not going to be very useful then because there's so few of them. Chances are you're mostly going to kill other things and the pesticide is not going to reach the one that you wanted. So sterile insect technique is really great for when you have a small population in the field and you can produce a lot and you can overwhelm it. But if the populations are already really, really, really high, then you're probably not going to be able to reach those ratios that you want. And you'll have to turn to other means, like could be pesticides, could be other areas of control.
So you're working in this fascinating space where genetics, ecology, and global health all collide. What do you think is coming next? And what do you see shaping the future of this field?
So there's a lot of effort being put right now into ways to make the sterile insect technique last longer and have males that are more fit, right? So moving beyond cobalt-60 based irradiation. So now we have access to genome editing tools where we can use to more specifically and more targetedly make changes to the mosquito or whatever fly genome you want, that we can encode sterility as a trait. And the sterility can be conditional upon maybe some cue that we provide. So we don't need a radiation source, and we know exactly what the change is going to be. And maybe combining that with some type of genetic system, because now we have all these genetic tools where we can say, okay, under these conditions, we can get males and females in the factory, and we change the conditions in the factory, and now all of a sudden we only get males and they're only sterile. And so these are happening. We have some really good techniques. There's a lot of really productive people in the field of vector biology and genome editing that are developing these transgenic strains that offer some really good promise to be able to scale up SIT in a way that it hasn't been able to before, because you can achieve sex sorting and you can achieve these kind of lethalities. I think there's a lot of attention being paid now to maybe having these guys be more fit, right? So coming out of the factory, having them you want them to be able to fly as far as possible, live as long as possible, make them as competitive as possible, and there's more attention being paid there. And then delivery is really changing, right? So if you think back 50 or 60 years, you're talking about a biplane flying over an area. Every once in a while, somebody pushes out a crate or a cardboard box, and then it crashes, hits the ground, and the flies fly out. It's pretty crudely done, and results in a lot of the flies dying in the process. And another reason why you need really high ratios. But now a lot of these are being delivered by drones, where drones can go to precise areas, and then they'll deliver and release small quantities of males in over a much larger area. So you don't need the males to disperse as much, right? If you have release a giant box and expect them to all fly 500 meters in every direction versus taking an army of 500 drones with a small amount and releasing them every 1 kilometer, you can cover a much larger area. You don't need them to fly as far, and you can get that same kind of coverage. So what we're seeing now is a lot of these mechanisms in both the genetics of how we make the strains, but also in the delivery systems of how those programs are delivering to certain areas. And if you tie that to monitoring, now we have these computer networks that can say, okay, this is where we're detecting the most flies, and then we change our releases here. And then, you're getting real-time monitoring data. And so the computational power lets us make these programs a lot more nimble, so they can change very quickly based on where the insects are showing up. You can change the number you're releasing in different places much more readily than you could have before.
So how do you communicate the value of this work to funders, to policymakers, and even the public?
Yeah, I think that you just have to kind of keep the problem alive, right? So people that remember what the problem was, right? These people have to be your strongest advocates, and we see that now with the screwworm as it resurges and has broken the boundaries at Panama and is heading northward again, and it's kind of rekindling a lot of the fears that it might return to the U.S. It hasn't yet, but there's a fear that it might, and we still have people that grew up in the 1950s and early 1960s who, maybe they were teenagers, maybe they were kids, but they remember seeing what that looked like and how that impacted their families and their organizations. And so these are your advocates to tell you what can happen and why we really need to be proactive. And they're the ones who really had the momentum to stop it in the first place, and they're the ones that are your advocates the second time. And we hope that the powers that be will listen to those folks and help protect them.
Okay, wait. They broke the border?
Yeah, a few years back, the barrier in Panama broke down. And they have been found in most of Central America now, and even into Mexico. And so there's a very large effort now to rescale some of the old production facilities or build new production facilities. And so the facility in Panama was meant to be a containment, to hold the barrier to produce, the 20, 30, 40 million flies per week. But it's not enough for eradication, right? So the old factories in Texas and Mexico that could produce 100 million per week, those had been shut down and shuttered decades ago. And so now there's an effort to restart some of those factories and get that production going again. SIT works, but it has to be at the scale to work. And so that's in progress.
Okay, last question for you. And I'm in awe right now because I have just learned so much over the last 45 minutes or so. This has been really educational and fascinating. But question for you, is there a person, place, or thing that has left you in awe recently?
I'm really interested in how genomes evolve, how they change, and how changes that occur over evolutionary time lead to new phenotypes and changes in behavior and whatnot. And I was reading this paper where this group found that when they looked at the genome sequences, because we sequenced a lot of genomes of different organisms, they looked at the genome sequences of primates, right? And so, to monkeys, to gorillas, to people, everybody sequenced all these genomes of primates and looked at this one region. They found this one region where all the primates that have no tail, like you and me, we don't have a tail. They had this selfish genetic element that had inserted itself in the middle of a gene. And none of the primates that still have tails had it. And they thought, well, that's an interesting correlation. And they took basically the same type of selfish element and they inserted it using genetic tools into a mouse genome. And mice, of course, have tails. And mice have the same gene that humans have. So they put it in the exact same spot and they made the exact same disruption. And what do you think happened? The mice lost their tails.
No tail.
And so we lost our tails because some selfish genetic element jumped into one spot and changed one of our genes just a little bit. So now we can't make a tail. And all it would take would be somebody to engineer that right back out of the genome again. And then we could get our tails back.
I'm mind blown by some of the things you told us. So very excited for our listeners to hear everything you have to say.
Thank you for having me on.
Zach, thank you so much for walking us through a world most of us never think about, and we're incredibly fortunate that we don't have to, and showing us how biology, behavior, and big systems come together in really powerful ways. Fascinating to hear the history of sterile insect technique, the scientists who thought up using nuclear science to begin with, just fascinating, and the incredible impact it's had. For me,
that is true. I am grateful for the access. And honestly, doing this episode has made me so proud to work in this industry. I am so proud of everything that nuclear does, but I'm still not sure I'm going to be eating a cheeseburger today, at least. Give me a couple days.
If you enjoyed this episode, make sure to follow Fissionary on Apple, Spotify, or wherever you listen. And while you're at it, please leave us a rating or review. It really helps others discover the show.
Thanks for listening. We'll see you next time on Fissionary.